Hello, welcome to Examining Origins. I'm here with Dr. Change Tan. She's a biochemist and she's done a lot of work looking into uh, things about origin of life and evolution with regard to prokaryotes and eukaryotes. And that's what we're going to be talking about today. Do prokaryotes and eukaryotes share a common ancestor? Welcome, Dr. Tan.
Thank you for having me. So do
Mhm.
you want to introduce this?
Yes. So I saw this image of the tree of life, and it shows that bacteria, prokaryotes, and eukaryotes had a common ancestor. What has your research shown about that?
Well, let's examine the idea whether bacteria, archaea, and the eukaryote, the idea that they share a common ancestor holds any water from the perspective of molecular biology. This actually is connected to, I think, a dialogue between you and your dog.
Well, it's
So,
not my dog, just a stray.
oh,
Yes, are you my mommy? Well, sorry, fella, we may have some similarities, but I can't possibly be your mommy. So, how does this relate to our talk today?
yeah, it's about whether prokaryotes and the eukaryotes could have shared the same mommy, right? That's the question. And we are going to address that from two perspectives. So one is molecular machines for DNA replication, transcription, and translation. The other is essential genes. So we'll see today that bacteria, archaea, and eukaryote, they have a very distinct DNA replication, transcription, and translation machinery. This I call organism-specific genetic information coding and decoding. So I'm going to illustrate my points using two analogies first. Do you like gift?
Yes, I love a gift.
You do. Well, that's because you are thinking in English term, right? That's GIFT means in English, it's a present. It's a present. However, if you are Germany, right, then that's a poison. I don't think you would love that.
Oh, wow. Okay. Good. Fun fact. Mhm.
If you ask a Chinese who does not know English and ask whether they want a GIFT, they'll say, what? What are you talking about? Because in Chinese, it's gibberish. So if GIFT is a code, then the meaning of the code depends on the decoding system. So if we are thinking in English, it's a present. If we are thinking in terms of German, then it's a poison, and in Chinese, it's a gibberish. So the meaning of the code depends on the decoding systems. Well, maybe now you don't want to answer my question anymore. What do you see here?
Not to be not.
Not to be not. That's because you started reading the sentence from the beginning to the end, right?
Yes.
And if I tell you that actually the sentence is cryptid, then depending, how are you going to decrypt it, what cryptographic key you use, that can be to be or not to be, or or to be not, or not to be not. All depends on where you start and where you end to read, right? The same words can mean very different things, even opposite things. So this actually is very similar to genetic information coding and decoding. So it seems that the inheritable genetic information that determines life or death of all living beings is encrypted. Not only that, they are encrypted in different ways in different organisms. Different organisms have their own unique cryptographic keys, and those keys actually are their own RNAs and proteins in their own cells present at the moment.
Wow.
So there are two very important aspects for genetic information coding and decoding. One is that it depends on the intimate cooperation among DNA, RNA, and proteins. Secondly, it's organism specific. Make sense?
Yes.
Wonderful. So basically, we needed to match the coding and the decoding systems. This is illustrated by this cartoon, which I do have a copyright to use. It's not made by me, but I acquired the copyright to use. So the function of an arrow really depends on, the function of the bow, right? They needed to work together. Separate, they cannot do anything, but together they can conquer the world. Okay, so another illustration of organism-specific genetic information coding and decoding actually is demonstrated in molecular cloning every day. And here I have an example of vendors making a synthetic bacterial cell. So that's the synthetic bacterial cell here, and the bacterial genome was introduced and cloned in a eukaryote cell, the yeast cell. However, the yeast cell, a eukaryote, cannot give birth to the synthetic bacterial cell. Just as, isolated DNA, you can have many copies of bacterial genomic DNA sitting in a test tube. They will never become a bacterial cell. That's because the eukaryote cells cannot use DNA replication, transcription, and translation machineries of bacteria to replicate or to transcribe or to translate its genomic DNA and vice versa. And we're going to see why. So first is the replication machinery. Here, I'm just showing you a comparison of DNA replication initiation in bacteria like E. coli on the left. Where's my mouse? On the left. And DNA replication initiation in eukaryote like yeast. So each of these symbols represent proteins, different proteins. Of course, it's very obvious that many more players are involved in eukaryotic DNA replication initiation. But the key thing here is not the number of genes involved or number of proteins involved, is their identity, right? The proteins required for DNA replication initiation in E. coli and in yeast are mostly unrelated. They are unrelated in sequence. Therefore, that actually, that is my beginning of my journey, thinking about the problem of the idea that all organisms have shared a common ancestor, because the genes involved in their essential processes, like DNA replication, transcription, and translation, are using mostly unrelated genes, right? Here, you cannot say, well, the bacterial one get more complicated, add more genes, so we have a eukaryote, because the eukaryote need their own genes to start their DNA replication. And you cannot say, oh, maybe life started with eukaryotes, right? And later on, the eukaryotes were simplified and streamlined so that we have bacterial. That could not work either, because the bacteria needs their own unique proteins to initiate their DNA replication. Yes.
So I noticed that, there's both sides have this protein, this helicase that divides the DNA, but they do look very different. And are you saying they come from a different gene completely? Like it's not even, there's no correspondence on the gene between this helicase and that helicase?
Right. So the only similarity they have is their helicases.
The function.
So the function is the same.
Mm-hmm.
They will open the double-stranded DNA to make a replication bubble. So the function the same. However, the bacterial helicase here is made of DNA B, so that is just a one protein, and that one protein will have you have 6 copies of them, and they form a ring. They behave like a helicase, unzipping the double-stranded DNA. The eukaryotic helicase is made of 6 different proteins. And each of those proteins is essential for the function of eukaryotic helicase. None of those helicase is related in ways the bacterial helicase. So basically you have bacteria and the eukaryote doing the same thing, but the implementations are different, right? They are using unrelated genes to do the same job.
That's amazing, isn't it? Even if
it is amazing. Furthermore, not only that, actually, you can see from the cartoon, right? For bacteria, you have origin binding protein, recognize the origin replication first, and that will start, the opening of the DNA double helicase. So making a bubble here, a replication bubble here. And the helicase will be then uploaded with the help of helicase loader. And the replication bubble will be enlarged, and then replication, can start. However, in eukaryotes, the recognition of origin of replication recognition complex does not create a replication bubble. And addition of the helicase, DNA helicase, cannot make a replication bubble either. And that's just a license, the orange of replication, and the licensed orange of replication needed to be activated to make a replication bubble.
How is it activated?
It's activated by a cascade of phosphorylation and the recruitment of many different components, many other proteins.
And that doesn't happen in the prokaryote.
No.
Okay, so we're seeing a major problem here, because if you have a prokaryote or a common ancestor, if it already has the ability to replicate, why would it create this other complicated system of replication? Is that
right? So basically, if we had the bacteria first, and then bacteria evolved into eukaryote, then it seems you have to change what is working in bacterial to make it not working anymore, so that will kill the bacteria, right? And before you have the eukaryote system, you cannot have eukaryotes. So basically, you have it's not like even between the bacteria and the eukaryote, it's not even death, because death means something used to be alive, right? It's inviability. You cannot have life there. Next, let's look at the transcription machineries. So here on the left is the RNA polymerase core of a bacteria. It's made of 4 proteins and with 5 subunits. On the right are RNA polymerases of eukaryotes. So bacteria only has one RNA polymerase. Does all the transcription. But eukaryotes, each eukaryote organism requires at least 3 different RNA polymerases. The simplest one is RNA polymerase 2, which is used to transcribe all the protein coding genes. And that is a polymerase actually most similar to the bacteria RNA polymerase. Yes,
Dr. Tan, often when we see these blobs, it really makes it look like this is just clumps of clay that are stuck together that could easily be rearranged. A protein, even the word, it doesn't really do justice to what it is. These are incredibly complex machines, and some of them are more complex than what we have even built in human machinery. So this is
much more complicated.
But can you explain what is more about what these little proteins actually are?
Okay, so these little blobs actually are derived from X-ray crystal structure of complexes. So different color represent different proteins. And each protein is made of many different amino acids. So each of those blob actually is a very complicated protein, where the core enzyme can make RNA molecule. They can, okay? If you give them a template, they can make an RNA. However, for gene transcription occur, you need help for other things. For bacterial core enzyme, it needs the help of the sigma factor. So that sigma factor is a bacterial specific. There's no sigma factor in eukaryotes. And with the core enzyme and the sigma factor, that together they will make the RNA polymerase holoenzyme. So that's an enzyme that can do gene transcription. We saw that eukaryotes, the simplest eukaryote RNA polymerase is made of 12 different proteins. Remember that?
Yes.
But the eukaryote RNA polymerase actually needs help from many, many different proteins. It's a lot. I don't have accurate counting, but you'll see. They need help from basal or general transcription factors, transcription activators, mediators, and chromatin modulators. So here, polymerase 2, which is made of 12 different proteins, is showing gray color here. It needs the help from 1, 2, 3, 4, 5, 6 different general transcription factors help to even have the basal level of transcription. And you can see that except transcription factor 2B, all other transcription factors are made of more than one proteins. The transcription factor 2D, for example, is made of 14 different proteins and 20 different subunits. Transcription factor 2H is made of 10 different proteins. So when you have all the general transcription factors together, the polymerase itself really becomes so insignificant. And that's just for basal level of transcription, right? For real gene transcription, you need help of many other things. Here is just a addition of mediator. So mediator itself, even though it's shown with just one single color, right? So on the left is front view, back, the right is back view. So this is just the polymerase itself, and this is a polymerase plus the general transcription factors, which are 2Ds, 2F, 2A, 2B, blah, blah. And the mediator, right? So mediator itself is made of more than 20 different proteins.
Wow.
So now let's look at bacterial and eukaryotic translation machineries. Well, here I just, I'm showing you a count of translation initiation factors. I restrained myself from showing you ribosomes, which are the molecular machines doing, linking the amino acids together to make a polypeptide bonds. The simplest ribosomes are made, of 50 different, more than 50 different proteins and multiple RNAs. But even though the ribosomes seems doing the job of putting the amino acids together, but they cannot start gene translation without the help of translation initiation factors. Bacterial has 3 translation initiation factors, so we can count them easily with 3 fingers. But if we want to count the numbers of eukaryotic initiation factors, we need to have a 6-fingered hand, right? I only have 5 fingers. And, most of our fingers would need to have branches, right? Actually, the numbers here are misleading because each of these translation initiation factors can be made of multiple proteins. For example, this initiation factor, eukaryotic initiation factor 3 is made of 6 to 13 different proteins, depending on the organisms. Two bacterial initiation factors have some sequence similarities to two eukaryotic initiation factors, 1 to 1A, 2 to 5B, right? However, those perform different functions in bacteria and eukarya. So here we have the same strand of RNA, and depending, whether the RNA is used for translation in bacteria or in eukarya, even if they can be used for translation, right? Because the way bacteria and the eukarya determine whether a segment or an RNA can be translated, and where is the translation starting and ending site. Different protein or no protein will be synthesized using that RNA at all.
Okay, so now I want to just make sure I'm understanding this properly. So in this one segment of the DNA of prokaryotes and eukaryotes, you have the same like nucleotide string, like you have these same letters, but this is where your analogy about gift comes in, where
Right.
it may have the same letters, but it means something different to the
Right.
prokaryotes and eukaryotes.
Exactly.
Okay.
So even if they have the same nucleotides, that's the point, okay? They can mean totally different things or actually has no meaning at all.
Okay, so but most of the segments of the code don't correlate. They don't have the same letters between prokaryotes and eukaryotes. Right. But in the two places where they do have the same code, it doesn't mean the same thing to each organism.
Right. So even if you have the same thing, for example, you can clone a human gene. You can clone a human gene into a bacterial. We can do that, right? And of course, we needed to do some tricks. And that's actually, I'm going to talk a little bit more in the next slide. So we can, clone human genes into bacteria, vice versa, yes. But for a human gene to be transcribed and to be translated in bacterial, you need to make the human gene look like the bacterial gene. So basically, you need you need to have a visa, right? When you go to the board to the customer, the only thing they read is your visa, right? If you have a valid visa, you're allowed to enter the country. It does not matter, whether you are Rebecca or Change. So you need to dress the human gene as if it is a bacterial gene, and then
how do you do that?
Will transcribe and translate it.
How do you do that? How do you dress the human gene like a bacteria gene?
Good point. Let's go to the next. So before we do that, actually, let me just mention briefly about archaeas, because many people believe that eukaryotes are sisters of archaea. So in the perspective of genetic information coding and decoding, that is DNA replication, transcription, and translation. Indeed, archaeas are more closely related to eukaryotes. But depending on what gene you compare, some gene will tell you where archaea are more closely related to bacterial, and a different set of gene will tell you where bacterial are more closely related to eukaryotes. So different genes will tell you different story. And even the same gene, compare different regions of the gene may tell you a different story about the relationship of different organisms. So anyway, just briefly, here I'm just showing you one of the subunits of eukaryotic RNA polymerase 2, that's RPB1. The largest subunit is called RPB1. And actually there are two homologs, two archaea homologs, RPBA prime and 2 prime, that can be aligned to RPB1, right? So they have some sequence similarities. So this is the alignment. So the green segments are the regions that the archaea protein and the eukaryotic protein can be aligned. So there are sequence similarity. Those purple marked regions are eukaryotic specific, and those green marked regions are specific to archaea. I just want to draw your attention to this C-terminal tail, which is specific to eukaryotic RNA polymerase 2. However, this C-terminal is necessary for RNA polymerase transcription initiation, elongation, and termination, and processing of transcripts made by RNA polymerase 2. In essence, that little C-terminal is essential. You cannot get rid of it and have a eukaryotic organism. So even, small parts of a gene can be the the question of life versus death, not to say, totally different genes, right? So now, we'll look at the molecular machineries for DNA replication, transcription, and translation. And we'll look at the essential genes. So I will answer your question about the cloning part, because that is a very, very the question. And I love that question because I do a lot of molecular cloning. So I put that in the end because it's so important and it's so cool. So let's look at essential genes first. Well, an essential gene is a gene in an organism that is necessary for the viability or the replication of that organism. Strikingly, that we have found that most essential genes are taxonomically restricted. So their distributions or the distribution of their homologs are very limited, right? Here I have a comparison of 6 different bacterial and 5 eukaryotes. You can see that the vast majority of bacterial essential genes are bacterial domain specific. So you cannot find things similar in eukaryotes. The eukaryote essential genes are mostly eukaryote specific. You cannot find similar genes in bacterial. So this really, suggests to me based on both what we know about the machineries involved in bacterial and eukaryote, DNA replication, transcription, translation, and also their other essential genes suggest to me that eukaryotes could not have evolved from prokaryotes. And this is actually demonstrated. It's demonstrated by molecular cloning on the daily basis. That's why you ask a very important question. And where I see that really based on what we know, life on Earth, different organisms on Earth cannot be linked by one big evolutionary tree of life, and not a web either, but a forest. So my working model is we have a forest of life with a similarity, discontinuity, and modularity, and the different life forms can share one or multiple modules. Okay, let's look at the molecular cloning. You'll see, I think you'll agree with me, molecular cloning is like an exhibition museum, right? Where exhibit the discontinuities among different organisms. So here I'm just showing you an E. coli and the yeast shuttle vector. So this is a cloning vector that can be used in both bacterial E. coli and in eukaryote, okay, like a yeast. So to be able to do that, we need a bacterial origin of replication, which can be recognized by bacterial to start to replicate the plasmid, the vector, right? To have the same plasmid to be replicated in eukaryotes, this vector also needs an origin replication that can be recognized by the eukaryote DNA replication machinery to start the replication of this plasmid, just a little plasmid, which is a short one.
Now,
yes.
In this diagram, are we trying to show a yeast being inserted into an E. coli, or a yeast gene, or an E. coli gene going into a yeast, or neither?
So this vector, this is a tool. It's like a car. You can use to do both, right?
Okay.
You can load cargoes. So the cargoes will be inserted in a MCS, that's a multiple cloning site. So you can clone here any gene. You can clone a bacteria gene here or a eukaryote genes, including human genes. You can clone here. But to make copies, to have this, plasmid, that pieces of DNA to be made in bacterial, to be replicated in bacterial, you need an origin replication for bacterial that can be recognized by bacterial. Because bacteria and the eukaryote, they have a different machinery and they have a different definition about whether something is an origin of a replication that they can use to start replication or not.
Okay.
Make sense?
Yes.
Okay. And also cloning, during molecular cloning, selection is essential. Otherwise, you cannot isolate or identify the cells carrying the DNA or genes, you have cloned or you are trying to clone, right? So for the selection in bacteria, we need bacteria normally is a bacterial antibiotic resistant genes. And the bacterial antibiotic resistance needs to be under the control of a bacterial recognizable promoter. So the promoter is a segment of DNA that will be recognized by transcription machinery. Because the differences between the prokaryotes and the eukaryote transcription machinery, a bacterial gene can only be recognized if they have a bacterial promoter, right? So a bacterial antibiotic resistant needs to be under the control of a bacterial promoter, so that gene can be recognized as a gene in bacterial. And we also need selection in eukaryotes. So you need to have a eukaryote-specific selectable marker. And here in yeast, is a commonly used marker is a nutrient selectable marker. For this gene to be recognized and as a gene, this gene needs to be under the control in a promoter, a eukaryote promoter that can be recognized by eukaryote. Because bacterial and eukaryote use different transcription machineries, they have their own way of determining whether a segment of DNA, even the same segment of DNA, right, whether it's a gene or not. Where they should start transcription and where they should end their transcription. So really you can see that where if you need something to work in bacterial, you need to make it that the bacterial can recognize as such. And likewise, if you wanted to make something in eukaryote, you need to make it so the eukaryote machinery can recognize as whatever you want it to be.
And you have to take those parts directly from the eukaryote itself or the prokaryote if you're doing bacteria. Mm-hmm.
Right.
So that's the visa. You have to bring something else over.
Right. That's you need to apply for a visa. So let's look at again the vendors making synthetic cell here because it's very illustrating, right? So for the bacterial gene to be cloned in eukaryote, they have to start with inserting an origin of replication into the bacterial genome. So the genome can be cloned in eukaryote. Right. However, even the whole genome has been, assembled and cloned in eukaryote, and you can make thousands of copies of the bacterial genome. But the eukaryotes, even though they have many more machinery and they have a much more complicated machinery, they could not interpret the code of the bacterial. That's why, the eukaryotes cannot give birth to a bacterial cell. Just as if, you only have the genomic DNA with no transcription or translation or replication machinery at all, right? Because they are just, they don't mean anything for the eukaryote. So the cloned bacterial genome needs to be transferred into a bacterial cell that can recognize the genes as genes, and the the replication, initiation site, those kind of thing, right? And even with that, even you can recognize all the genes, you have all the machinery match, missing of one essential gene will kill the whole project. So vendor has the misfortune of had one deletion of a nucleotide when an essential gene of the bacterial. And that actually made them unable to make the synthetic bacterial cells until they fix the mistake. So essential gene is essential, it's required for of the organism. You cannot miss even a single one. And even the simplest life form we know of needs hundreds of essential genes.
And for those watching who may not be familiar with what you mentioned, you're talking about the experiment with Craig Ventnor where he wanted to reproduce, he wanted to make a synthetic cell, but he didn't really, it's not as a lot of times it's promoted as if he created life from scratch, but all he did was take parts from other cells and then make a cell together, right?
Exactly, yes. So that's why even though they said that they created a new species on the earth that has a computer as its parents, but I call it molecular cloning because it's clone. It's a copy, yes.
I have a question about that. So, are you saying, because I don't know about that much about cloning, are you saying that we can take a prokaryote, like a part of a prokaryote, we can put it in to the eukaryote and make basically a clone, but it can't reproduce? Is that?
Well, we can clone a bacterial gene into eukaryote, and that gene can be replicated and can be transcribed and can be translated. For example, LAC-Z gene is commonly used as a marker in eukaryote. It's an E. coli gene, right? We can clone one gene from one organism to another, but you need to clone in a way. If you just want to replicate the DNA, then you just needed to clone it using a vector that can be recognized and replicated in the host cell. Or you can insert by some cloning trick, insert the bacterial gene into the eukaryote genome. Again, you needed to have the eukaryote promoter, those kind of thing, to have that bacterial gene to be recognized as a gene and to be transcribed and to be translated in eukaryote. So we can do the cloning. We can clone genes from one organism to another. But you needed to do a trick to basically dress your cloned gene, apply a visa, right? You needed to dress your cloned gene as if, so it looks like the host gene.
Okay.
The host gene, the host does not care the interior, what is the inside of the gene, right? What is the inside? They only care the ends, the promoter, that's a transcription, that will tell the cell where is the transcription starting site. And you need a transcription stopping site to tell the organism this is where you should stop transcription. And you also need to manipulate the gene to clone it in a way. Basically you need to make a chimeral, right? A chimeral, so you dress the gene not the middle, but the ends, so that your host will think your for your host, basically your for your host, well, this is your gene. Okay, this is your gene. So that's why actually we can clone a toxic gene. We can clone a toxic gene into an organism. If you dress the gene correctly, then the organism will think it's a good gene and will transcribe and translate the gene, and then will be killed by the product it made itself.
That's very fascinating. Some people, I feel like they will say, okay, well, yes, the eukaryote is much more complicated, and the prokaryote and the eukaryote, they took different paths toward replication. But, it could still have a common ancestor because the common ancestor was very, very simple. And so maybe it had some very simple way of replicating that we no longer have today. So how do you respond to that?
Well, since replication and the transcription and the translation are essential, right?
Mm-hmm.
And if the common ancestor has that system, then all its offspring, needs to use similar system to replicate their genes and transcribe their genes, translate their genes. Otherwise, you cannot, you cannot break the bridge, right? Because they are so different, the genes involved has no sequence. Most of the genes have no sequence connection. And the only connection we can have from parents to offspring is through DNA replication, right? Replication will basically pass on the genes.
Mm-hmm.
So the only way of doing that is basically where inherent our genetic information was sequence inheritance.
Mm-hmm.
It's not functional inheritance, it's a sequence inheritance.
Yes, I see.
So you needed to have that, you always needed to have that functioning. For any organism to evolve into another, that organism need to be able to stay alive and to replicate or propagate, right? Or reproduce.
So there's no time to redo your whole system of replication, right? Like you don't have that kind of time because you need to be able to replicate every time.
Time depends on how fast you can do things. Yes.
Mm-hmm.
So if genes can pop out easily, right? Like formation of crystals, sodium chloride salt crystals. If they can pop out very easily, then yes, doesn't matter how many different genes you need, how many essential genes you need, because you can just pop them out, then doesn't matter, right? The question is, all we know of, so many people actually studied how easy it is to generate a new gene, or how easy it is to make a functional gene remain functioning, and to guess how easy it is to generate a new gene. So basically, in all the sequence space, what is the possibility you have something functioning like a gene? The answer, in short, is that accumulated mutation nobody has observed experimentally, right? Any accumulated mutation could generate a brand new gene. We can make chimeras of genes by ourselves. So that's molecular cloning. You can make chimeras, but we don't have the ability to generate a brand new genes.
Wow. Thank you so much, Dr. Tan. This has been amazing. Appreciate your time.
Thank you so much for having me. It's fun to talk with you.
Yes, and if anybody's more interested, you can look at our other talks with Dr. Tan. The links are in the description of this video. We did one on abiogenesis, and I hope to do more in the future. Thank you.
Thank you. Take care.